Right. So today's topic, from where we left out in the last class, students, was immunity. Okay. So we will carry on with this. Okay. Just 1 second. Okay. Just 1 second. I will just get back to you. Give me a second. All right. Yeah. So in the last session, we were talking about quite an important things about immune system, right? So while we were discussing about immune system, I told you that immune system is not just 1 second. Uh, yeah, all right. So immune system is not just one particular, it's not confined to one particular organ or anything. I told you it is a very elaborate system. You remember? So while I was explaining that, I told you that immune system can be an organ. I think somebody has please mute. Please mute the call. PPT not visible. Okay, great. Now it should be visible, right? Yeah. So let's go back to our session. Yeah. So in the last class, I was talking about immune system, and I told you the brief introduction to what is the importance of immune system. And while we were discussing that, I also told you that an immune system can be a culmination or a combination of several different organs, tissues, cells, right? In our body, they integrate together to give you this defense mechanism. And in that topic, we specifically talk about lymphoid organs, primary lymphoid organs, secondary lymphoid organs we discussed, and tissues and cells and antibiotics. We're going to talk about it in the coming session. So we will discuss about the same thing again by the end of the chapter, and we will revise this. Okay. So let us head on to what we have to talk today. All right. That is immunity. So what is immunity? We know that immune system gives us defense, right? Immunity is a mechanism of how the immune system performs to give us a function. Performs to perform this particular function. So if you talk about immunity or the mechanism of immune system, we come across with two different types of immunity. One is innate immunity, other one is acquired immunity. So the immunity in definition is the ability of our body to distinguish between the self cell and the non-self cell. So self cell is something that is within us. For example, our own tissues are called as self cells. And non-self cells are something which is not belonging to our body, which we call it as antigens. So this capability of our body to distinguish between these two comes in two different categories. One category is something your body doesn't know what it is defending, but it gives an overall protection. Like, for example, a lamination. You don't know what you're protecting it from, whether it is heat, whether it is rain, whether it is an ink. You don't know, but you give a proper coating so that nothing affects it. So this is called as non-specific immunity. I don't know my enemy, but I want to just be sure that nothing enters. That is called as non-specific immunity. And the other part is specific immunity, where your body deliberately knows what it is attacking. It plans strategy, it has memory, it knows what kind of an army it has to call upon to neutralize this thing. So, based on this difference, that is the general immune mechanism to a normal immune mechanism, it is called as innate immunity and acquired immunity. So, let's talk about it. When we talk about innate immunity defines as non-specific immunity. So as I said, what is non-specific defense? I don't know my enemy. Simple. I don't know my enemy, but I'm going to act as a barrier. I'm going to act as a difficult portion for any of the outer things to enter our body. Simple as that. So under this non-specific immunity, they are given by by nature, it's like from birth itself, you have this defense mechanism. So innate immunity exists from the time of the birth. Okay. So what are the types of innate immunity we can come across? One is physical barriers, physiological barriers, cellular barriers, and cytokine barriers are some of the different types of immune barriers with respect to innate immunity we can come across in our body. So let us discuss each of these barriers, and then let's get familiar with these terms. Now, if I talk about innate immunity, under innate immunity, if I talk about physical barriers, okay, so physical barriers are very observable, and you can just understand its role very simply by just looking at your hands or legs or your face. What do you see when you look at these? You can see that our body is covered by a proper layer of skin which protects all our internal organs like muscles, blood vessels, tissues, bones, etc., and the internal fluid. So now this physical barriers, one of the major example we can pick up is skin, one of the largest barriers that we have in our body. What do skin protect you from? Skin protects you from heat, light, dust, antigens like microorganisms. Okay, it can be bacteria, it can be fungi, it can be virus or a protozoan, right? Or it also protects you from the external. It completely separates your body from the external environment to the internal environment. So that is one of the major roles of your skin. And your skin also has a natural defense where it secretes some oil, right, through sebaceous glands. So you can see that on your face, you can see there is a greasy substance that is a sebaceous secretion that is coming from your oil glands in your skin, and it also secretes, sweat. So I know that the sweat is produced due to the exercise or because of the external heat to balance the body temperature. But if you see the sweat, the properties of the sweat also provides us some kind of a defense mechanism. So sweat is known to have this antimicrobial properties that will prevent any of the microbial infections, especially from the external environment. So that is about skin. Now coming to another type of physical barrier is mucus coating. While we were studying about human physiology, and especially we talked about digestive system and respiratory system, and even in the third chapter that when we discussed about human reproductive system, one thing that we observed is the tracts of gastrointestine or gastrointestinal tract or urinogenital tract. Okay, so or the respiratory tract, it is lined by a special type of an epithelium where it produces mucosa. You have gustatory or gastric mucosa, you have respiratory mucosa, and you have urinogenital mucosa. So all these mucus secreting cells or the goblet cells that are present, they secrete a special or a unique type of this mucus for pertaining to a particular type of an organ or a tract where they trap these microorganisms and they have antimicrobial properties where they digest this entire thing. I can give you one example. The genital tract of a female secretes mucosa which is rich in lactic acid. So the lactic acid present in the genital tract of a female is antimicrobial in nature. So it prevents a large amount of any of these antigens entering in our body. All right. So two types of examples that we discussed in physical barriers. One is skin, other one is mucus coating. Right. So these are the physical barriers. Coming to the physiology or the physiological barriers. So it's very easy to understand the difference between physical and physiological. When we talk about physical barriers, it is physical, it's just plain. Physiology is something when your cells are performing a specific function and they perform a specific activity that is called as a physiological barrier. So, in physiological barriers, they also do the same job of preventing microbial growth without having a specific goal of what microbe. It's very non-specific. That's what I mean to say. In physiological barriers, we have the examples such as gastric HCl, saliva, and tear. So this might be very, very familiar. When we talk about the HCl present in your stomach, the HCl present in your stomach is very acidic. We know that the pH of the stomach is around 2 pH. Right? The pH is around 2. It is so acidic that it can really melt an iron bar. Why do we have such a concentrated acid that is present within our stomach, which is like a really, really harmful thing to carry around? We have this for two reasons. One is the HCl in the stomach helps us break down the protein. And also, the HCl in the stomach neutralizes 98% of the microbes that are entering in our body through the food and the water we intake. We know that the majority of the infections that we suffer from come from food and water. So that is why we have to give a large amount of credit for the gastric HCl or the HCl that is present within your stomach. And the next one is saliva and the tears. The saliva actually also contains the lysozymic enzymes that are antimicrobial in nature. These lysozymic enzymes do not have the capacity to digest your tissues within your mouth because your mouth and the oral cavity and the esophagus is very well covered by the mucus. So this mucus gives you a protection against the action of the lysozymic enzymes present in the saliva. Whereas the external agents who are entering into your mouth through food or the air, what happens is they are prone to the action of these lysozymic enzymes from the saliva, thereby eliminating them or digesting them. And the next part is the tear. We hate tears, right? We hate to cry. We always regard tear to sadness. So is it all the function of the tears? Definitely no. The tears also have antimicrobial properties. It protects the most sensitive organ in our body, that is the eyes. Always the tear glands keep on secreting these tears with lysozymic properties such that no fungal or bacterial or viral infections occur on the surface of the cornea. That is why the tear glands always moisten your eyes and keep your eyes wet so that no infections occur during this course of time. So under physiological barriers, we understood that we have three different examples. One is the gastric in the stomach, the saliva in the mouth, as well as the tears from the eye. So next time you get a tear in your eye, remember that they also help in your eyes to get a protection. Okay, so that is about that. And coming to the next slide, cellular barriers. So the cellular barriers are like a patrol or a police that are just moving around the tissues, the cells, etc. They have a very unique capabilities. We know that human beings have a closed circulatory system. So if these are our blood vessels and the blood vessels bifurcate to become smaller and smaller capillaries, eventually they do not have the capacity to open up. So all the blood components, they do not escape these blood vessels, so they remain within the blood vessels, right? But our phagocytes, or for example neutrophils or natural killer cells, they are amoeboic. For example, you have a macrophage, amoeba looking like cells. So they squeeze within the cells of the capillaries and they come out of this blood vessel. To do what? They patrol around the tissues and the cells in our body and they look for any possible antigen that is hidden or gotten into our body. So they engulf this pathogen like an amoeba engulfing its prey, and then they digest it using lytic enzymes that they have within their lysosomes. Similarly, we have other kinds of cells such as neutrophils. Remember, the other name of neutrophils is also called as polymorphonuclear leukocytes. So leukocytes stands for white blood cells. Why do we call it as polymorphonuclear? You can see that the nucleus, which is into small lobes, they come in different shapes and structure. That is why it is called as polymorphonuclear leukocytes. So they are also non-specific in nature. Next comes monocysts and neutrophils. Monocysts and natural killer cells are another three examples of cellular barrier. So their job is very simple. They patrol your body and the blood vessels, and then they look for any possible antigens. If they recognize any possible antigens, they just attach to this antigen and they digest them, neutralize them, and then kill them. Okay, so this is about the cellular barriers. Coming to cytokine barriers. So cytokine barriers are very, very unique kind of barriers. So what happens in cytokine barriers? So basically, let us assume that we have a cell and the cell is infected by a virus. When a virus infects the cell, the infection begins by virus attaching to a cell. If this is the virus and this is the surface of the cell, the virus comes and attaches to the surface of the cell, and within the virus, there is a special package, and this package we call it as virus capsid. So this capsid or the package of this virus contains its genetic material and the proteins or the enzymes that enable this virus to survive within your cell. Once it comes and attaches to the external surface, the virus injects its capsid into our cell. So now what will happen? The capsid opens and the DNA gets integrated within our DNA, therefore taking control of our whole cell. Now, during this process, the virus wants to make more and more of its own viral proteins or viral RNAs. So they start producing more and more of the viral RNAs or the DNAs through transcription. Okay, through transcription process where it produces RNA, and then reverse transcription where RNA to DNA happens. And each of the DNA molecule that they have synthesized, which are called as viral particles, they will be released as new viruses, and they attach into a new cell, and then again you have new infection cycle. So now what happens is some of the cells, they have the capacity to detect the detect the viral DNA presence, and they produce a special type of antiviral proteins that degenerate the viral particles or the viral proteins. So once they degenerate these viral proteins, now these cells, okay, which are the non-infected cells which were able to detect this virus, what they do is they actually release some signals. Okay, they are able to pick up some signals that are produced by your infected cells. So these infected cells, when they release these signals, they are called as interferons. So the interferons are the special type of signals that are traveling from the infected cells to non-infected cells. The non-infected cells pick the signals. With that signals, it produces some antiviral proteins, thereby killing the viral infection or stopping the viral infection. So these are called as cytokine barriers. All right. So our next topic is acquired immunity. So before I go to acquired immunity, I want to make sure if you understood what I just thought. Okay. So give me 1 second, students. All right. I hope it is Venkat Rao. Are you able to follow? Right. So no problem. If you have any doubts, please put it in the chat box. Okay. Yeah. And the next topic is acquired immunity or specific immunity. So our body has a very unique capabilities to recognize which is its enemy and which is not its enemy. So when it is able to specifically look at an enemy and thinks that, okay, this is from the outer, this is an antigen, and I have a specific defense mechanism to it. So that immunity is called as an acquired immunity. As the name suggests, acquired immunity does not come by birth. Acquired immunity is built through the course of your lifetime. That is why we are just encouraged as children to go out and play in the soil, swim in rivers, lakes, okay, and encourage being as environmental as possible and just get mixed up with the surrounding environment. As we become more social with the environment, now what will happen? We are introduced more and more to the antigens that are present in the soil, in the water, in the air, wherever you live. So when you're constantly exposed to these antigens, sometimes you develop fever, sometimes you develop infections. But gradually, as you grow up, you develop a very strong immunity towards all these agents during the course of lifetime. So this building up the capabilities to prevent any possible infection because you already have an experience of it. That is called as an acquired immunity. So this is also called as pathogen specific immunity because it knows what it is doing. Okay. So if I have to understand my defense, if I know that I have to attack someone, so that means I should know who I am fighting. If I have to know that who I am fighting, definitely I should have memory, right? So if I have, for example, if somebody beats me and goes, the next time I should, if I have to attack them or if I have to prevent them from attacking me, that means I'm able to recognize them. So if I'm able to recognize them, that means I have a memory of it. So that is called as, okay, a memory based immune response. So they build a memory. So when did I build a memory? When I got first beaten up. So when I got first beaten up by someone, that is called as first encounter. So when I have my first encounter, even though I'm hurt, what I'm doing is I'm able to remember that this is the people that attacked me, or these are the things that infected me, or these are the things that has affected my body. So that is called as first encounter. The first encounter is the first time you get infected, and your body learns about this infection in a very not so convenient process. Okay. Now, once you have the first encounter, your body produces something called as primary response, or it is also called as low intensity response. When i when my body has to fight someone which is unknown, now it doesn't know how to just defend itself. So what it will do, it will give a very generalized response. Okay, overall response. Okay, something that can help, something that can prevent. So this is called as very preliminary response or low intensity response. Sometimes the pathogen that has entered may be affected or may not be affected. So this is called as low intensity or preliminary response. Now, once a preliminary or low intensity response is done, it will take a longer time to get rid of this infection. For example, when you are a child and when you had a fever, it would take several days for you to heal yourself. Why? Because your body did not know how it is trying to, combat this disease. It would take a very long time to understand the dynamics. Now, when you're grown up, hardly you would suffer for a day or a two, and you would be perfectly all right. How did this happen? Now, once you had this preliminary response, your body understands the type of antigen that is coming in your body, the ways to recognize it, and the means to combat it. So once it has understood this dynamics, now the secondary encounter becomes very easy. In the secondary encounter, the moment the pathogen enters, it produces a high intensity response. It is called as an asthmatic response or high intensity response. So once it produces an high intensity response, it specifically knows what it is doing or who it is attacking, what kind of an attack it has to produce. So that is called as high intensity response. So now these both primary and the secondary responses are carried out by a special type of cells. And these cells are called as B lymphocyte cells and T lymphocyte cells. So these are called as lymphocytes. When we talk about B lymphocytes give our body an ability to recognize our antigens by producing something called as antibodies. We'll talk about what are antibodies. Antibodies are simple proteins. So these simple proteins are designed in such a way that they go and attach to the surface of your antigen. For example, if a bacteria is coming, the way the bacteria is recognized by our body is somebody going and attaching, and telling them, "Hey, this is bacteria." So that job is done by these antibodies. So antibodies are produced in such a way that they are produced based on the memory of the past encounter. Our past encounter gives the messages to these B lymphocytes to construct certain protein based on how it remembers it. So the B lymphocytes produces antibodies such that Based on the information, it goes and recognizes a specific part of this antigen, whether it is a glycoprotein, whether it is a flagella, anything. Okay. So once it recognizes this, then comes T lymphocytes. T lymphocytes are also called as cell mediated immunity, or it is also called as C cells. So these T lymphocytes, they are responsible for cell mediated immunity based on the messages of the B lymphocytes. What they do is they uh, they go and attack this particular pathogen, producing specific number of immune cells, producing specific type of immune cells for that specific antigen. Understood? So, 1 second, students. All right. So for us now, we have to go on with studying the structure of antibodies. So I just mentioned you a few seconds ago that the role of the B lymphocytes is to produce something called as antibodies. And these antibodies are nothing but special type of proteins. So proteins are made up of polypeptide chains. We already know. So our antibody, the structure of antibody is very, very important. Okay. Our antibody is made up of 4 polypeptide chains. And among these 4 polypeptide chains, 2 are called as identical light chains and 2 identical heavy chains. That's why we represent it as H2L2. H stands for heavy, L stands for light. So H2L2. So there are several types of antibodies which are called as immunoglobulins. Ig stands for immunoglobulins. Immunoglobulins. So based on that, we have IgA, IgG, IgM, IgE, IgD. Example, IgA is found in mother's milk, right? You know that IgA is found in mother's milk. IgE is responsible for allergies or allergic reactions. So these are immunoglobulins that play a role in giving a particular type of immune response. Chalo? So when we talk about the structure of an antibody, it is made up of 4 chains. So these are called as light chains and these are called as heavy chains. Both these light chains and heavy chains, you see, a light chain and a heavy chain is held by one disulfide bridge. A light chain and a light chain is held by 2 disulfide bridges. And here, a heavy chain and a heavy chain is held by 1, 2, 3, 4. Okay, 4 disulfide bridges. So basically, these light chains and heavy chains are connected together by disulfide bridges. Okay, so one thing that is very unique here is the variable region. What are these variable regions? The variable regions which are present at one certain region of your light chain and heavy chain are the ones that go and recognize these antigens and bind to them. Understood? So these are called as antigen binding sites and they're also called as variable regions of heavy chain. So these are the regions that recognize your antigen and go and bind to them. So please memorize the entire structure of your antibody. It's very, very, very important for your examination. For example, heavy chain, disulfide bridges, okay, where the bridges are present, okay, what is constant region, what is variable region of light chain, and what is variable region of heavy chain. So these learnings are quite important, okay. And next we'll talk about what is epitope and paratope interactions, okay. So I think we're short of time. Okay. Okay. We will end here and we will take up in the next class. All right.